Now the question from the year IJSO 2016 related to protoplasm is, if the DNA was made up of 6 nucleotides instead of the 4, then what will be the total number of triplet codons possible? We have already studied that the mRNA sequence, it consists of 4 nucleotides on nitrogen bases, that is AUGC. And with that we were saying that we need to make a triplet codon, we will need around 64 codons will be possible to code for 20 amino acid. So here also we are saying that instead of that 4, we are saying that the nucleotide that are present are 6. So here we are saying that, for example, if the nucleotides they are present in 6, then to make the triplet codons possible, we'll say that we'll need the 6 to raise to the power 3, so to make the triplet codon possible, that is the 6 nitrogen bases, they have to come in a triplet form so that they can code for the amino acid, because here we are having the nitrogen bases, that is they are made up of 6 nucleotide sequences instead of 4, so that's why here we are writing it at 6, and we want the triplet codon. So if we try to make the combination, then we will get around 216, 6 into 6 36, and again 36 into 6, that will be equal to 216. So if we are saying that instead of that 4 nucleotide sequence of AUGC present on the mRNA, if we have the 6 nucleotide sequence, and from that 6 nucleotide sequence we want that again triplet codon like AUG, GUG and so on. So from this we are saying we will get around 216 possible codons which will code for the amino acids. So the answer for this question will be D, that is 216 will be the total number of triplet codon possible if the nucleotide sequence is made up of 6 nitrogen bases. So now the next question, again from the same year 2016, states that which of the following is true about ATP? ATP stands for the adenosine triphosphate. So this ATP, we have to find out the statement, that is which among the following is completely true with respect to the ATP. So firstly, it is a derivative of one of the nitrogenous bases that form DNA. Yes, completely it is the correct statement that it is a derivative that is being formed from one of the nitrogen bases, that is adenine. Adenine that is found in DNA. We have talked about the 4 nitrogen bases that are present in DNA, that is A, T, G, C. A stands for adenine, T for thymine, G for guanine and C for cytosine. So here we are saying in ATP, A stands for adenosine and it is a derivative compound derived from a nitrogen base, that is adenine. And this adenine is also found in DNA, therefore we are saying that yes, it is a derivative of one of the nitrogen bases that also help in the formation of DNA. So the first statement is correct. Second, it splits into ADP and phosphate and the energy produced is used by the muscle cells to contract. Yes, it is definitely true, that is the energies required by the cell is in the form of ATP. Now to utilize this ATP, it is being broken down into ADP, that is adenosine diphosphate plus phosphate group. So this amount of energy is being used also by the muscle cells to contract and to show movement. So yes, we can say that yes, it splits into ADP, that is adenosine diphosphate and a phosphate group, and this energy can be used by the muscle cells to show contraction. It is produced in both aerobic and anaerobic condition. When we talk about respiration, we say that there are 2 types of respiration. One is the aerobic, other is anaerobic. Aerobic, which occurs in the presence of oxygen, it also produces ATP and large amount of ATP is being produced. And anaerobic condition, in which we are saying it does not require oxygen, or that kind of respiration which occurs in absence of oxygen. It also produces ATP but in less amount. So we'll discuss about this in later chapter, that is the respiration, but giving a brief idea here, we are saying that yes, ATP it is produced in both aerobic conditions and anaerobic condition. So seeing all the 3 statements, we can say that yes, all the 3 statements are true and the answer will be D, that is all of the above statements are true with respect to the ATP. So now the next question from the year 2017 IJSO related to protoplasm is, what would be the minimum required length of codon to code for 400 amino acids if the condition is that there existed 3 purines and 3 pyrimidines each in number? So overall they are trying to ask that what will be the triplet, that is we were having the triplet codon to code for the 20 amino acids. But now they are saying that, for example, we have 400 amino acids, and in that one more condition they have stated is that we have total number of 6 purines and pyrimidines, that is 3 purine and pyrimidine each in number they have told. So total number of purines and pyrimidines are 6. So with respect to that, we have to identify what will be the length of the codon so that it can code for 400 amino acid. So if we try to calculate it by putting each and every nitrogen base, then it will be a very tedious task because it will take a long time to calculate. So there is a short trick, or we can simply say that when we calculate the length of codon, in general we were saying that we were having 4 nitrogen bases. When we were actually talking about the codon sequence which is accepted universally, we were saying that we were having these 4 nitrogen bases in the mRNA, that is AUGC, and we have to identify the length of codon stated as n, and they should almost be equal to code for the 20 amino acid. So this was our condition, and with respect to this condition we got that when we have placed the power, or when we have given the value of n as 3, we were saying that we were able to code for 20 amino acid because 4 raised to the power 3 was giving us 64 codons possible, which will be sufficient to code for these 20 amino acid. So applying this condition, we can get the answer of this, in which we are simply saying that we have to code for 400 amino acids. So for example, here we are saying that we have to code for 400 amino acids. So the number of codons that should be possible should be greater than or almost equal to, so that they can code for these 400 amino acids. And here we are saying that the total number of purines and pyrimidine present are 6. That is, they have given 3 purines and 3 pyrimidine each in number. So the total number is 6. We have to simply identify this length of codon. That is, we have to simply identify the length of, or the value of n. So if we try to place the value of n firstly as 1. For example, if we place the value of n as 1, then 6 raised to the power 1 will be highly insufficient to code for 400 amino acid. If we place the value of n as 2, then 6 raised to the power 2, that will be equal to 36. Again, they will not be sufficient to code for the 400 amino acids. Then we are saying that if we place the value of n as 3, that is, we are saying 6 raised to the power 3, then the value will be 216, again insufficient to code for 400 amino acid. But when we place the value as 4, that is 6 raised to the power 4, we get the value as 1296, which is more than sufficient to code for the 400 amino acid. So the answer to this question will be 4, that is the length of the codon required to code for 400 amino acid, in which we are also having purine and pyrimidine number equal to 6, then the value of the length of the codon will be 4. Now to another question again from IJSO 2017 related to protoplasm is, when heated, the hydrogen bonds between the complementary DNA strands break, and these strands ultimately separate in the process known as the melting. So they are saying that which piece of DNA will require higher temperature for melting. So simply we are saying that we have found out a link, that is the hydrogen bonds present between adenine and thymine, they are double hydrogen bonds. Whereas the hydrogen bonds present between guanine and cytosine, they are triple hydrogen bonds. So more temperature will be required to break the triple bond between guanine and cytosine as compared to break the double bond between adenine and thymine. So here we have to simply see the number and the amount of GC content found in the DNA. If the GC content is more in the DNA sequence or that strand of DNA, then obviously more temperature will be required to break that strand as compared to the other strands of the DNA. So simply we have to see the amount of GC content found in the sequences. So as you can see in the first sequence, we are saying this is the GC bonding. Again, we will simply highlight the GC bonding in the DNA strands. So we are having 1, 2, 3. Simply 3 GC bonding is present between the first strand given in the option A. If we try to see the option B, we will simply see the amount of GC content. So here it is 1, 2, 3, 4, 5, 6 and 7. So here we are having 7 pairs of the guanine cytosine triple bonds. When we try to see the third option, in third option we will mark the GC content. Here 1, 2, 3, 4, 5, 6, 7, 8 and 9. So here we are having 9 pairs of the guanine and cytosine. And when we see the last option, or when we mark the GC content, here we are having 1, 2, 3, 4, 4. So when we try to see the amount of GC content in all the 4 strands of the DNA, the maximum amount is found in option C. So maximum temperature will be required to break these strands of DNA as compared to the other strands of DNA. So the answer for this option will be C.